Well, good morning. I'm going to be covering some of the same ground that Steve did, since I'm talking about adoptive transfer. But whereas his presentation really focused on an approach that you might call intelligent design, where you look at the targets that you're supposed to be using the immune system to recognize, I'm going to describe an approach that makes use of the evolutionary capacities of the immune system. Now, I've spent many years in Texas, so I accept that it's quite important that intelligent design be incorporated, but I think evolution has positives. Okay, so as you appreciate, the adoptive cellular therapy can really apply to almost any cell in the immune system. But I'm going to focus, like Steve, on T cells, partly because they're the most broadly used, but also because their characteristics are such that it makes them very appealing. In particular, their ability to recognize internal antigens, unlike antibodies, their ability to traffic through multiple tissue planes, ability to self-amplify and recruit lots of other effector mechanisms, reducing the risk of resistance, and also, as I said, their ability to evolve with the tumor. And that's critically important because, of course, solid tumors vary in a single individual, not just in time, but in space as well, so that it's very difficult to come up with a universal antigen. Now, T-cell mediated cancer therapies have been extremely effective, whether you use the T-cell's own native receptor or an artificial chimeric antigen receptor. But I think it's true to say that with the exception of melanoma, the most striking successes have been with B-cell tumors. So what I thought I'd do is ask why the activity is so high with B-cell tumors, and how can we learn from that and extend the approach to solid malignancies. And I'll begin with native receptors, and native receptors directed on T cells to Epstein-Barr virus, a virus that infects 90% of the population, is lifelong latent in almost all of us, expresses viral proteins throughout our lives, but is usually pretty benign. In a proportion of people, though, it produces Hodgkin or non-Hodgkin lymphoma, which contains and expresses some of these virus-associated antigens. It can also induce nasopharyngeal cancer, which is a common tumor, particularly in South Asia. Now, the people who really suffer from EBV-associated malignancies are people who are severely immunocompromised by disease or by iatrogenic causes. And up to 25%, for example, of stem cell transplant recipients get a very aggressive immunoblastic lymphoma. So a long time ago, about 20 years ago now, we thought that if we could put back functional T cells that recognized Epstein-Barr virus antigens into these patients, we should be able to get rid of the antigen-infected cells and therefore destroy their tumors. And we genetically marked the cells so we could track them in vivo, and really found, I think, some fundamental features of T-cell therapies, which are that you get very rapid, very extensive expansion in vivo, 10,000-fold in just a very few days, that you get long-term persistence, more than 10 years, and in more than 120 high-risk patients, none of them got the disease. Importantly, though, 90% plus of the patients who had established immunoblastic lymphoma showed a complete and sustained response. How well do these work in people who've got a normal immune system and should apparently have their own Epstein-Barr virus specific T cells circulating? Well, not quite as well, but nonetheless, in patients who have relapsed or resistant disease, again, more than 50% have a complete and sustained remission. And over the past few years, we've been improving on the techniques in terms of making them simpler and more robust. And we've gotten very consistent results with that approach. This has now been taken up for clinical use trial by biopharma. So why did these work so well? Several reasons, really. First of all, we're targeting strong and unique and consistently expressed viral antigens, which are presented with ample co-stimulation. As you heard from Jay, you have to have multiple signals to ensure that a T cell gets through all its various checkpoints, and normal B cells and certainly antigen-presenting cells expressing viral antigens do have on their surface all these accessory signals, so that once you've engaged the target, the T cell receives all these additional signals and is able to proliferate and persist. And this sustained immune response, importantly, allows the immune system to evolve its response to the tumor. So if you look at the pattern of immune response after you've administered virus-specific T cells, you start to find the appearance of other tumor-specific T cells in the circulation. T cells that you didn't put in, but that weren't there or weren't detectable previously, but have now become evident. And if you actually take these same specificities and give those directly back to other patients with EBV positive or negative lymphoma, you can start to see tumor responses. So these are contributing to the immune responses that we see. Well, although T cell receptors are kind of attractive and native T cells are appealing, there are some disadvantages of the T cell receptor. Antibodies, for example, can recognize non-protein tumor antigens, and they're MHC independent and unrestricted. And that's important because many tumors are negative or don't process MHC negative or don't process antigens very well. So again, about 20 years ago, Zelig Eshar developed a concept of combining the properties of a T cell and a monoclonal antibody, taking the antigen binding component of a monoclonal antibody, coupling it to the signaling domain of the T cell receptor, introducing it into a T cell, which would then recognize its target through the antibody, but would be able to retain the desired properties of the T cell, that is to say trafficking, expansion, persistence, and effector function, but would now be able to target a broader range of antigens. When these were first tried clinically, and this really explains the 20-year gap between introduction and success, they were effectively inert. These so-called first-generation chimeric antigen receptors produce incomplete activation and limited killing. It was only when we introduced into these receptors co-stimulation, secondary signals, that allowed a single engagement to produce both signal 1 from the chimeric antigen receptor and signal 2 from the co-stimulatory molecule, that we started to see improved T cell activation and proliferation and killing of tumor cells. And I think, as most people here will know, that's been spectacularly successful for the treatment of CD19 positive malignancies, in particular for acute lymphoblastic leukemia, now more than 90% response rates in many, complete response rates in many centers, and even for lymphoma, better than 50% response rates. And that's directed to the CD19 antigen on normal and malignant B cells. I'm not going to show many clinical pictures, but I do want to emphasize that this approach can remove even massive tumors that are resistant to other therapies, and it's a single dose administration and prolonged responses. This has triggered multiple large-scale biopharma studies, and what's interesting is that chimeric antigen receptors directed to other B-cell antigens are looking also very promising, albeit in much earlier phase studies. So what is it that's special here again about B-cell tumors? Well, it's a strong and it's a unique antigen that's presented in the presence of ample co-stimulation. If we try and see what happens in solid tumors, however, we have very few antigens that are strong and unique and consistently or stably expressed, irrespective of whether they're neoantigens or not. Instead, we have an evolving pattern of antigen expression. And what's more, the tumors lack co-stimulation. But it's worse. There's not just passive ways in which solid tumors are less attractive than B-cell malignancies. They also have an actively hostile tumor microenvironment. As Jay pointed out, there are masses of cytokines and cells and adverse chemokines that render the environment very hostile to any immune response that might develop. So what we considered was that we might be able to use viruses and virus-specific T cells as a means of addressing both those problems. So what we do to begin with is use T lymphocytes whose native receptors are virus-specific as a platform for chimeric antigen receptors and get the critical co-stimulation in trans. What I mean is we have a virus-specific T cell expressing a chimeric antigen receptor directed to the tumor. Its native receptor directed to a virus. It sees the virus on a professional antigen presenting cell, gets all necessary stimulation and co-stimulation through that, and in its fully licensed state, then traffics off and kills the tumor through its chimeric antigen receptor. Now, we tested whether this kind of concept could work in patients with neuroblastoma, a pediatric malignancy, and we targeted it to the pediatric malignancy with GD2, which is a ganglioside antigen expressed at high density. We started out using a first-generation chimeric antigen receptor, no co-stimulation. So remember, this has been shown over many years to be essentially inert in any clinical setting. And indeed, when we give that to patients who don't have a virus infection but have neuroblastoma, so they're only getting stimulated through their chimeric antigen receptor, we see essentially no expansion. When these cells, however, same cells, go into a patient who's got an active virus infection that will stimulate them through their native receptor, then we see massive expansion, 10,000-fold expansion, persistence over many weeks and months, and the disappearance often of quite bulky tumors shown here in the bones. The problem is this is a completely impractical approach to cancer therapy. You can't wait until the cancer patient develops an overwhelming virus infection before you treat them. But what you can do, perhaps, is to substitute a planned oncolytic virus for a random systemic viral infection. So the idea is you take peripheral blood mononuclear cells from a cancer patient, induce virus-specific T cells using our fairly rapid and robust approaches that we've developed for EBV, but this time directing it to the oncolytic virus, transduce it with a chimeric antigen receptor, for example, to GD2, and then reintroduce it into the patient who receives an oncolytic virus vaccination and then gets in vivo expansion as planned. Now, the idea is that you don't just use a naked, as it were, oncolytic virus, but you use a virus that contains immunostimulatory or immunomodulatory genes. There's already one such virus licensed, TVAC, which contains GM-CSF. You might consider this a first-generation oncolytic virus, but second- and third-generation viruses are able to contain a multiplicity of these genes. For example, a chemokine to attract T cells to the site of the tumor, a cytokine to activate them once they're there, and single-chain antibodies to produce checkpoint blockade. In this way, you turn a cold tumor, an inhibitory tumor, into something that looks to the patient like a hot virus infection. You transfer your T cells that target the virus in the tumor, and you develop a potent and, crucially, an evolving immune response to solid tumor antigens. Now, each of these components works well clinically. Each of the components in combination works well in animal models, but I wanted to spend the last 5 or so minutes describing why it's been so difficult to get this translated to human use and what the issues are really that will, at our present rate of progress, as Samir says, take us 300 years to test all these different combinations. The problem is that as you go from biological therapies that are relatively simple, like non-viral vectors, towards one that become increasingly complex, to the kind of totally individualized therapies that Steve was describing, or the combination therapies that I've just described, the similarity to the conventional pharmaceutical model progressively declines. And it declines because these therapies are often individualized, even if some of the components are off the shelf. Their complexity means that you can't follow the standard drug development pathway from preclinical, phase 1, phase 2, phase 3. Instead, you're locked into an iterative phase between preclinical and phase 1, where you test out your best product, go back to the lab, improve it, back into the clinic, and so on. That's extremely difficult for any commercial entity to entertain that kind of open-ended time and money commitment. The third component is that there's multiple components in any complex biologic therapy, which gives a requirement for multiple licenses to be agreed. And although that's not an insuperable problem, it's a very, very slow one to overcome. And the last, but by no means least problem is it's a totally different economic model. The basic model for pharmaceuticals, I guess, is that you spend a lot of money up front developing an approved drug. You then manufacture it for a few cents or fractions of a cent per pill, and then you administer it ideally lifelong to a patient in whom it's ameliorative and for whom you can charge the maximum possible amount. That sounds cynical, but that is the sort of fundamental commercial model of most drugs. These drugs, however, are ideally one and done. They're curative, given a single time, but they're very expensive to make, and they always will remain expensive, even as costs come down. We don't know really how to price them or how to get the cost recovered. So why does this matter? Well, it matters because if you look at the development of standard drugs from preclinical discovery through preclinical validation, phase 1, phase 2, phase 3. What happens in general is that academic involvement diminishes over time. And it diminishes over time because the costs increase logarithmically. And you really have to get involved with an entity that has far more financial muscle than an academic. If, for the reasons that I've just described, you look at the development of complex biological therapies, then academics have to remain involved a lot longer. And because academic funding tends to diminish with time, certainly very difficult to get funding for the crucial but slightly tedious development phase of any drug, what that means is that both the academics and certainly their products enter the notorious and much feared valley of death. So how do we overcome that? I don't think there's any one single way of doing it, but I'll just tell you briefly how we've tried to do it as a sort of a starter for discussion. We've developed a center that has basic translational and clinical components so that we can go from development, from sort of discovery through to the development phases. And why that's important is because it allows us to obtain programmatic funding. And programmatic funding allows you to cover the costs of these kinds of early phase 1 studies. It doesn't allow you to cover the costs of phase 2 studies, and it's at that phase when we've de-risked the product, as it were, when we've done these kind of iterative phase 1 studies, that we can form partnerships with biopharma to take it forward. We don't know the best way of doing this. We've tried 3 different approaches. We've tried broad-based collaboration where a whole space or field or whatever you want to call it is licensed. We've tried licensing individual products for fairly narrow pivotal trials, and we've tried developing things actually in-house. Each of these approaches has got its own pros and cons, but I would say that 3 elements that are shared by all of them and that need to be addressed, they're not impossible, but they're concerns, are first of all a culture problem. I always used to think that culture was a sort of very soft word and didn't actually mean much, but it really can. And to put it sort of crudely, it's the culture of collaboration versus the culture of confidentiality. Again, there's nothing pejorative about what I'm saying. It's just that it is different and people think differently in different settings. The publish or perish versus perish if publish. The second problem is a more recent one, but it's troubling me more and more, I have to say, which is that the clinical outcomes now of very early phase clinical protocols are having massive financial implications. The valuation of various companies can change literally by billions of dollars on the results of single patients almost in single phase 1 studies. That makes it very difficult for any academic institution or investigator who's involved with one of these studies to really be clear that they have no conflict of interest. It's manageable, of course, but it's a consideration that's a very important one. And the third factor, I think, is that the lack of cross-licensing opportunities can lead you to use suboptimal combinations. In other words, you're not going to go forward with a combination that you know is not going to be licensable from other commercial entities because there's no point in it. You're not going to get beyond phase 1 or phase 2. And that, I think, is something that also needs to be considered and addressed. Finally, and something I'm not going to discuss in any detail, is that the post-licensing economic models. How do we pay for these very expensive one-off therapies? And I think some of the approaches that hopefully will be discussed during the meeting are alternative approaches to approval, alternative paths to approval, reallocation of resources from the kinds of incomplete therapies that are currently used that need a lot of patient sustenance and the kind of hospital industrial complex, as it were, to support the failure of current therapies. And of course, and I think in my opinion, most crucially, excellent health services research, which is done much earlier in drug development than is normally the case and justifies the expenditure. So I think I've finished just on time for you. And thank you for your attention.